I was starting a collaboration called Genome 10K, which was a view to bring together people in different fields. Genomics people, mathematical analysts, and biologists who were basically picking up rocks to find snakes and frogs because they needed to talk to each other, because we need the animals to do the sequencing. We need the genomics people to understand how to interpret the genomics, and we need the mathematicians to be sure we don't make mistakes. When I came here and I talked to the leaders of BGI, I said, we need your help. You have this technology. You can help by producing sequences which we can help you analyze, but more important than analyze, interpret in a biological sense, in an evolutionary sense, in a medical sense. That's what's important. And that there's a lot of things that are happening despite the fact that people are trying to say, oh, well, genomics, it's old news. We need something new, and et cetera. But that's not true. What's true is that genetics and genomics, but the word genetics is part of every field of biology, and it just keeps getting better. In fact, 30, 40 years ago, medical schools didn't have genetics departments because they didn't know it was important. Then they did. Eventually they'll go away because they'll be part of every department. I see a lot of improvement in the last 10 years in the breadth of collaborations that have been encouraged at the highest level, because pedigree analysis, which was using families to discover genes, cannot be used in complex diseases. So they use the tools of population genetics to uncover distortions in gene frequency in people that get a disease versus people that don't get the disease. And that has led to some kind of discoveries that predict things like Alzheimer's disease, or HIV/AIDS infection, or diabetes risk. And those discoveries often are translated into drugs. If we can figure out how to disentangle the real hits, the real discoveries from the statistical artifacts, we should try. There have been major technological changes and major advances and major ideas and computer technologies and bioinformatics that have made discovery easier. They have made analysis easier. They have made analysis faster and made translation to the bedside quicker for medical purposes. All of those things have happened. But an area that is really just getting started now is what I call comparative genomics, where we actually have a number of genomes sequenced and analyzed by BGI and by genomics companies and laboratories across the world. And what they do is they're so good at producing a genome that has very few errors and has basically a way to look at it. So what I think will happen is more and more derivative analysis will be coming out from young scientists who we should encourage, whose head is not filled with all the junk our